Good morning, everyone. Hope you're all doing well. Hope everything is going good with other classes. And as always, if you have any questions or concerns, please email or leave a comment on Google Classroom and I can get back to you as soon as possible. Today, we're going to do two things. We're first going to review parts of the last assignment, and I have a new document and assignment for you. Okay, so please make sure you're keeping up with the assignments. The due dates are spaced out quite a bit, and that's to make sure you are not, you're not stressed out about any of this stuff. But if you can, submit your assignments sooner than the due date. That way, it's off your plate and you're on track. All right, so let's go over the last assignment that had been posted on Google Classroom. This is the natural selection introduction. All right, so let me grab the pointer. Okay, so antibiotics versus bacteria. Actually, let me pause for a second and I'll be back. All right, so antibiotics versus bacteria. This section we were talking about, or you had to define antibiotics, bacteria, and penicillin. There was a video about who Dr. Fleming was and what he discovered. So he discovered penicillin, which is an antibiotic. So the prefix anti meaning against, and biotics are anything living. And so bacteria are these living, one-celled, single-celled living organisms. And what he found, if you watch the video, is this fungus, penicillin, that would kill the bacteria. All right, so hopefully you got that. And if you didn't, now's your chance. Hopefully you can fill this out and turn it in. Okay, what is MRSA? I'll leave that up to you to watch the video and answer that question. Okay, then we had a link to a presentation. The presentation, I've opened it in the tab right here. It has an extra what do you call it? Slide in it now. So if you want to go back and look at it, that might not be a bad idea. Okay, so you were supposed to make a prediction. So the presentation, if you watch the video, it tells you that this mega plate, this is something scientists prepared. So it was a plate with food for bacteria, for E. coli, and they had sectioned the plate into different sections. And onto each section, they poured different concentrations of antibiotic, which is the killer of bacteria. And so what they were trying to see is how the antibiotic or the different concentrations of the antibiotic had an effect on the bacteria. And then what they saw, and you were supposed to predict what you thought would happen as the bacteria multiplied naturally, because that's what bacteria do. When given food, they will reproduce. And so as they move down the plate, or if they move down the plate, what would happen to the ones in the middle that has a super concentration of antibiotics? And what the scientists observed, so you were supposed to make a prediction. Okay, and this is an image of what they saw. The light colored or the cloudy colored sections are colonies of bacteria, and as they move down the plate, so at the very ends you have bacteria in agar, or the solution, the food, with no antibiotics. So the bacteria multiplied with no obstacle, and as they went down the plate, you can see the color gets lighter and lighter, or darker and darker. The black shows through the white. In the middle, you have the least amount. Okay, so another representation. Okay, so you have generation one, first generation of bacteria, and as they move down the plate or towards the middle of the plate, you get different generations. So in the middle, you get this generation, the red generation of bacteria. And so what is special about these bacteria in the center, and what the scientists found, and what you could tell just by common sense, is that these bacteria had to survive, had the genetics, right? This colony is not the same as this colony. It's like the, let's see, the great 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 grandchildren of the original colonies at the ends of the plate. So as the bacteria move down, right? So the dark blue bacteria didn't have to encounter any kind of antibiotic, any kind of poison. As the light blue bacteria crept into or was produced along the next highest, or the first concentration of antibiotics, only the bacteria that could survive, that had the genetic mutation to survive that poison, could go on to give birth to the, looks like puke yellow color, right? And so that went on until the bacteria in the middle are progeny or products of bacteria that had mutations to survive even the highest concentration of antibiotics, right? So these in the middle are the most dangerous, because E. coli, let's say if you get an infection of E. coli, you would be treated with antibiotics. But if you got infected with this kind of E. coli that's been exposed to antibiotics over generations and has survived, none of the antibiotic concentrations would work on this E. coli, and this would most surely be problematic, probably deadly. So these things in the middle we call superbugs, because they have kind of overcome a lot of poison, a lot of obstacles to survive, and so they have these defense mechanisms. Okay, so we can go further into this later on. It's very interesting, but in the interest of moving forward and keeping things short, we'll move on. I'll just tell you the defense mechanism is actually coating around their cell, so kind of like a shield that prevents the antibiotic from penetrating inside in the cell and destroying the cell. All right. So moving on to what we're doing today. All right. So we're gonna explore viruses now. So remember, bacteria and viruses are different. Bacteria are living most of the time single-celled, or actually all the time single-celled organisms. Viruses are kind of in the middle of living and not living, and you're going to explore that in this article. It's on Khan Academy. Let's see if I can open it. Now, you don't have to log in. This link should work for anyone. Some people see I've logged in, but you don't have to log in to access this article. Okay, so what you're gonna read about is, or what you're gonna learn about is many different kinds of viruses that infect humans and other animals and causes serious illness or cause serious illness. Okay, there's an introduction. What does an animal virus look like? Okay, that one should look really familiar given what we're going through right now. Okay. How do animal viruses infect cells? Okay, the mechanism, it's really, really interesting and very elegant. Viruses are actually the way they infect cells, they're vectors. So this mechanism is actually being used by scientists to infect you with medicine, right? So they're taking the shell of the virus, and then when you read what viruses do, they're actually taking out the disease DNA from them, DNA or RNA, and then putting in things that actually heal you. Okay, so that'll be interesting. Consequences of an infection gives you an overview of what can happen from different types of viruses: herpes, adenovirus, rhinovirus. All right. And then there's the classification of animal viruses, and I'm sure you've read in the news that the the current virus has been traced. Its origins have been traced into bats, and um, actually, this type of virus, coronaviruses, almost always come from bats. So that's the one clue that scientists have, that this virus has animal origins. Then there's a passage about the retrovirus HIV-1. Okay, this is the commonly known as AIDS, or the virus that causes AIDS. Okay, so by the way, the novel coronavirus, which is what this virus is named, causes COVID-19. Okay, that's the correct way to say things. Okay, so here's a diagram of the life cycle of a virus. Okay, and it tells you about drugs that um, work on AIDS, on the AIDS virus, or the virus that causes AIDS. All right, so that's kind of like an outline of what the article is all about, and the questions you have to answer. Please use your own words. You know, it's not hard to tell when you copy and paste. Uh, it's an awesome skill to learn, but hopefully you can, you know, hone your other skills in thinking and generating or synthesizing your own ideas. All right, so please use your own words. You can write it in paragraph form, which means more than one sentence. Okay, so how do animal viruses infect cells? The answers you can find. There are 3 consequences that result as a result of a viral infection, and if you're confused about where you can find that, I'll show it to you right now. Okay, so the answer is in these 2 paragraphs. There are 3 consequences, okay, that 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 are a result of a viral infection. So explore that and jot down a few original sentences about it. Okay, and then you need to summarize the life cycle of the HIV virus. And again, the passage and the diagram will give you more insight into that. All right, so please work on this. This will be posted by the time you watch this video on Google Classroom. And if, again, as always, if you have any questions or concerns, please email me or leave a comment on our classroom page, and I will get back to you as soon as possible. All right, hope you're all having a good week, and I will talk to you later.